Hi, hello, good morning. This is Ramesh Kumar Lalkota, Assistant Professor of Botany, MBS Government Arts and Science College Autonomous, Mahabubnagar, Telangana State. In our last class, we studied about translocation of organic solutes. Today, we will see respiration. Okay. So, what is respiration, how it occurs, where it occurs, what are the organelles in which the respiration occurs. We'll see all these things. Okay. So before that, all organisms require energy to carry out all the life activities. Okay. So all the living organisms continuously perform some life activities. So to carry out these life activities, what do they require? They require energy. Okay, so where do this energy come from? The energy comes from the food that we take. Okay, from the food that we take. Okay, so what is the ultimate source of energy? The ultimate source of energy is the sun. From the sun, the plants absorb the radiant energy and they convert them into the chemical energy. Okay, this chemical energy is stored in the form of organic compounds, and these organic form compounds, which are uh, prepared by these plants, form food for different other herbivorous organisms. Okay, so as the plants are autotrophers, they prepare their own food, and all other organisms which are heterotrophers, they depend upon these plants for their food. Okay, so this food, uh, in whatever type it is stored in the organisms, is it undergoes oxidative catabolism to release the energy, and this energy is utilized to perform various life activities. So this oxidative catabolism is nothing but respiration. In the presence of oxygen, the reserve food material or the stored food material is oxidized and liberates energy, and this energy is used by by all the organisms uh, to carry out the life activities. Okay. So uh, during this process of respiration, many other intermediate compounds such as amino acids and different types of other uh, intermediary compounds develop in the path of respiration, and these are used as starting points for synthesis of amino acids, aromatic compounds, pigments, and several other organic substances. So, and this process is known as intermediary metabolism. Okay, so respiration is very important. It not only oxidizes the reserve food material, and also the intermediary compounds which are formed in the pathway of respiration are used as starting points for the synthesis of pigments, amino acids, the aromatic compounds, and several cellular constituents. So this is about the introduction of respiration. The next part is the history. Okay. So, generally, people think that respiration as it's a synonymous word to breathing, that is the process of inhaling and exhaling. But plants do not perform this inhaling and exhaling, but they do perform respiration. So this concept of, that is, the exhaling and inhaling is not the right definition for respiration. Okay, then uh, Malfigi was the first person in 1679 who identified that seeds require air during their germination. Okay, seeds require air during their germination for uh, that means they do respire and convert the reserve food material into energy, and this energy is utilized for germination. Okay, so this was first found by Malfigi in 1679, that seeds require air during their germination. Then Dutrochet was the person who observed that the oxygen of air enters into the intercellular species of leaves and other, and from there into the cells for use in respiration. So he's the first person who observed that oxygen is observed during respiration. Then Lavoisier, what he stated that respiration is a very low combustion of uh, reserve food material, similar to that of burning of coal in air. Then comes Jan Ingenhousz in 1779. He showed that all living plants give out carbon dioxide. Okay. Then de Saussure in 1779, uh, based on the experiments of Jan Ingenhousz, he showed that the volume of oxygen observed by germinating seeds was equal to that of the carbon dioxide produced during their germination. And Sachs in 1885, uh, he considered a respiration as an oxidation process with or without the use of oxygen. If with use of oxygen, it is known as aerobic respiration, and if respiration takes place in the absence of oxygen, then it is known as anaerobic respiration. Okay, and Meldrum, in his work called Cellular Respiration, he emphasized that respiration is a chemical reaction by which organic compounds are burnt by oxidative reactions inside the living cells. Okay, so in the light of our present day knowledge, the respiration process includes oxidation of organic compounds either partially or completely. If partially oxidized, we call it as a fermentation process, and the end products formed are ethyl alcohol and carbon dioxide. And if they are completely oxidized, the process is known as aerobic respiration, and it takes place in the presence of oxygen, and the end products are carbon dioxide and water. Okay, so this is the comparison between respiration and photosynthesis. See, you can see the equations there. Respiration, how we write the equation for respiration: C6H12O6 plus water in the presence of oxygen gives rise to carbon dioxide, water, and energy. Here, the substrate is the glucose. C6H12O6 is the respiratory substrate. It, in the presence of oxygen, undergoes oxidation, and the end products formed are carbon dioxide and water, and energy is liberated in this process. Okay, 686 kilocalories of energy is liberated by oxidation of one molecule of uh, this glucose. Okay, how we write photosynthesis equation? See here, carbon dioxide and water are the reactants. Carbon dioxide from the atmosphere and water from the soil are observed, and these, in the presence of sunlight inside the leaves, that is in chlorophyll, these inorganic compounds are reduced into organic compound, that is glucose molecule formation takes place here. Okay, so here C6H12 O6 is formed, and here it is oxidized. It is the reactant here, and in photosynthesis it is the end product. In respiration it is the respiratory substrate or the reactant. Okay, so you can see here in respiration oxygen is observed, O2 is observed in respiration, whereas in photosynthesis oxygen is liberated. Okay, and CO2 is evolved as a result of oxidation of carbon compounds. This carbon compound, that is glucose, is completely oxidized in the presence of oxygen, liberating carbon dioxide. So here carbon dioxide is liberated, but here in photosynthesis carbon dioxide is a reactant. It is the inorganic compound which is reduced to glucose. So carbon dioxide is observed, and it is fixed inside to form carbohydrates. And respiration occurs throughout the day, that is irrespective of the time, that is it occurs throughout the day and night, and the light is not at all required for respiration to occur, whereas photosynthesis, it occurs only in the presence of light. What happens in respiration? The chemical potential energy, the chemical energy which is stored in the form of potential energy, is converted into kinetic energy here. But here in photosynthesis the radiant energy, that is the sun's energy, the radiation, is converted into chemical energy and ultimately stored as potential energy. Okay, here the potential energy is converted into kinetic energy, but in photosynthesis the radiant energy is converted into potential energy and stored. Okay, and the respiration, it does not require chlorophyll, but photosynthesis requires chlorophyll. Without chlorophyll photosynthesis won't occur. Okay, and the raw materials for respiration are glucose and oxygen, whereas the raw materials for photosynthesis are carbon dioxide and water. Okay, and respiration is an exothermic process. It is a catabolic process. What is exothermic? Heat liberating process. Heat is generated here. Oxidation of this glucose liberates a lot of energy, and some of the energy is liberated in the form of heat. Okay, so this is the exothermic process, and it is a catabolic process. Catabolic, what is catabolic process? Here the substrate undergoes a breakdown. Okay, the larger organic molecules break down to smaller. These are inorganic molecules, carbon dioxide and H2O. It's a catabolic process where breakdown of larger molecules is seen, whereas in photosynthesis it is an endothermic process and it is an anabolic process. Here heat energy is observed during these reactions, and it's a constructive process. Okay, it ends up in the formation of organic compounds from inorganic compounds. And this respiration occurs in cytoplasm and mitochondria. The process of glycolysis occurs in cytoplasm, and the other cycles such as Krebs cycle and electron transport system takes place in mitochondria, whereas photosynthesis occurs in chloroplast. Okay, and the respiration results in loss of weight. Photosynthesis results in weight gain, and during aerobic breakdown of one glucose molecule, 36 to 38 ATP are formed, and during synthesis of one glucose molecule, 18 ATP are utilized. Here they are formed. Here ATP are utilized. So these are the differences between the respiration and photosynthesis. Now we will see the respiratory apparatus. Where does this respiration occurs? Respiration occurs in cytoplasm as well as in mitochondria. The process of glycolysis occurs in cytoplasm, and the remaining processes takes place in mitochondria. So the function of mitochondria is to generate adenosine triphosphate, that is ATP, by oxidizing the respiratory substrates in the presence of molecular oxygen. Okay, in the presence of oxygen, what happens here? The respiratory substrate completely gets oxidized and liberates energy, and this energy is stored in the form of ATP. So these mitochondria are also known as powerhouses of the cell. These mitochondria are the site of cellular respiration. Each and every cell performs the respiration because all the cells require energy. This reserve food material is translocated to all the cells, that is throughout the body. Each and every cell receives the energy, and this energy is oxidized completely in the mitochondria of the cell, and ultimately energy is liberated, and this energy is utilized for various life activities, that is all the metabolic activities whichever are performed within the cell occur using this energy. Okay, so um, these mitochondria are typically rod shaped. You can see here they are rod shaped, and they are about 0.2 to 1 micrometer in diameter and 3 to 10 micrometer long. Okay, 0.2 to 1 micrometer in diameter, and 3 to, say, diameter from here to here, and they are 3 to 10 micrometer long. This is the length. Okay, so structurally these mitochondria are composed of two membranes. The outer membrane is smooth and the inner membrane is convoluted. Okay, the outer membrane forms uh, uninterrupted outer boundary of mitochondria, and it bears a number of spherical particles which contain NAD linked dehydrogenases. See, this outer surface has some spherical particles, small spherical particles, which may range up to 60 angstrom wide. And these particles, which are spherical in shape, they contain NAD linked dehydrogenases. Okay, they contain NAD linked dehydrogenases. And the inner membrane is convoluted. What is convoluted? Inner foldings. It shows inner foldings, finger-like foldings, to form a large number of cristae or ridges. Okay, which project into the inner chamber. See, you can see here clearly these are finger-like structures. In convolutions are infoldings. This is the inner membrane, this is the outer membrane. See, this is the outer membrane and this is the inner membrane. Inner membrane shows number of convolutions or inner foldings. Okay, these are called as cristae. And the space between these two membranes, that is inner membrane and outer membrane, is known as intermembranal space, and it is 80 angstroms in width. Okay, the intermembranal space, you can clearly see here, this is the outer membrane, this is the inner membrane, and the space in between these two is known as intermembranal space, and it is 80 angstroms wide. Okay, then uh, mm, and inside this inner membrane, you can see it is filled with a matrix. The inner membrane is filled with a matrix which contains dense granules, ribosomes, and mitochondrial DNA. The granules consist of insoluble inorganic salts and are believed to be binding sites of divalent ions such as magnesium, calcium 2 plus, magnesium 2 plus. Okay, so this is the structure of the mitochondria. And one more thing, um, uh, The inner membrane and the cristae are seen to be covered with small projections, finger-like projections, small, very small. These projections are known as F1 particles or ATPases or F0 F1 particles. These are spherical in shape, ranging from 8 to 9 nanometer in diameter. Okay, connected to the inner membrane by a short stalk. You can see in the diagram, small round particles are seen. Okay, these particles are known as F0, F1 complexes. You can see here. See, this is F0, F1 particles, the small spherical bodies, F0, F1 complexes. Okay, so they are also known as ATP synthesis, and they are involved in the coupling of oxidation and phosphorylation. And these F1 particles, uh, each F1 particle consists of a basal piece called a stalk and a head piece. The headpiece is 75 to 100 angstroms in diameter, and the stalk is about 50 angstroms in length. Okay, so this is the structure of mitochondria. So you can see here clearly, these are the F0, F1 particles on cristae. Okay, so this is about the structure of mitochondria. Now, cellular respiration. So cellular respiration is divided again in this respiration is divided into two types, that is aerobic respiration and anaerobic respiration. Aerobic respiration takes place in the presence of oxygen. Here glucose undergoes complete oxidation, and the end products are carbon dioxide and water. So in this, the first step is glycolysis, and um, glycolysis is a common step in both aerobic and anaerobic respirations. It occurs in cytoplasm. Okay, so glycolysis is the common step, and based on the availability of oxygen, if oxygen is available, then oxidative respiration, that is the aerobic respiration, continues. And if oxygen is absent, then anaerobic respiration or fermentation takes place. Sorry. So we will see the differences between aerobic and anaerobic respiration. Aerobic respiration. See, aerobic respiration is common to all living cells. Anaerobic respiration, it is common in only certain microorganisms. How? What is the equation? See, here in aerobic respiration, the respiratory substrate in the presence of oxygen, it undergoes complete oxidation, resulting in the formation of carbon dioxide and water. And 686 kilocalories of energy is liberated during aerobic respiration. In anaerobic respiration, the respiratory substrate is partially oxidized, and the end products are ethyl alcohol and carbon dioxide. Here you can clearly see that no oxygen is present. Aerobic respiration takes place in the presence of oxygen, but here no oxygen. So partial oxidation takes place, and a very small amount of energy is liberated. That is 56 kilocalories of energy is liberated during anaerobic respiration. And uh, aerobic respiration releases more energy per unit substrate consumed. That is, for one molecule of glucose, 686 kilocalories of energy is liberated. And here, for one molecule of glucose, only 56 kilocalories of energy is liberated. Okay. And aerobic respiration occurs in the presence of oxygen. This occurs, anaerobic respiration occurs in the absence of oxygen. Here, carbohydrates are oxidized completely into carbon dioxide and water, and here they're partially oxidized, and the end products are ethyl alcohol and carbon dioxide. Here, glycolysis, Krebs cycle, and electron transport system operate in this process. Here, glycolysis alone takes place. Krebs cycle and electron transport system, they doesn't occur. Okay. So, aerobic respiration, it is the usual method of respiration which takes place in the presence of oxygen. And with the help of enzymes and oxygen, uh, the respiratory substrates are completely oxidized to carbon dioxide and water. And the reaction is an exothermic reaction. As we said, that the energy is liberated in this reaction. Some of the energy is also lost in the form of heat. So this is an exothermic reaction. It's also known as exergonic reaction. Okay, and anaerobic respiration, here oxidation of respiratory substrates in the absence of oxygen is termed as anaerobic respiration, and it involves the incomplete oxidation of organic food into ethyl alcohol and carbohydrates, and only a little bit of energy is liberated. Okay, so this is about the uh, differences between aerobic and anaerobic reactions. Now, the next part of the lesson is glycolysis. What is glycolysis? Where it occurs? Who observed? Who identified? Who discovered this glycolysis? We will see. Okay, as I said earlier, the complete oxidation of reserve food material, that is aerobic respiration, takes place in four steps, four stages. Okay, the first stage is glycolysis, and it takes place in cytoplasm. And in glycolysis, the glucose is break down into glucose to pyruvic acid. The second reaction is the oxidative decarboxylation of pyruvic acid to acetyl coenzyme A. This reaction is called a link reaction between glycolysis and Krebs cycle. Okay? And this acetyl coenzyme A enters into Krebs cycle. Krebs cycle takes place in mitochondria. And the last stage of this Krebs cycle, this aerobic respiration, is electron transport system and oxidative phosphorylation. We will see one by one. Today, we will look into glycolysis. Okay. So, as I already said, glycolysis is a common path between aerobic and anaerobic respiration, and it takes place in cytoplasm. And the sequence of the reactions involved in glycolysis, first identified by Emden, Meyerhoff, and Pernas. So, uh, this pathway is also known as EMP pathway. Okay. Now, all of them are German scientists, and they worked between 1912 and 1935. The process of glycolysis is described taking glucose as a starting point, and it includes two major steps. That is, one is the conversion of glucose into fructose 1,6 bisphosphate, and splitting of this 1,6 fructose 1,6 bisphosphate into two molecules of pyruvic acid. See, glucose is converted into fructose 1,6 bisphosphate. This is the first stage, and this fructose 1,6 bisphosphate splits into two molecules of three carbons, ultimately results in the formation of two molecules of pyruvic acid. So we will see the reactions one by one. The first reaction is the phosphorylation reaction, where ATP is utilized. See, ATP is utilized, and the glucose uh, It undergoes phosphorylation in the presence of the enzyme hexokinase, and phosphoryl group is transferred from ATP to glucose, resulting in the formation of glucose 6 phosphate. This is the first reaction, phosphorylation reaction, and this reaction occurs in the presence of hexokinase enzyme. And see here, the glucose is phosphorylated at sixth carbon, so we write it as glucose 6 phosphate. And in the next reaction, this glucose 6 phosphate is isomerized to fructose 6 phosphate, and this reaction takes place in the presence of phosphohexoisomerase. This is isomerization reaction. Okay, glucose and fructose are isomers. Okay, so this is a isomerization reaction. And the see uh, this isomerization is nothing but conversion of aldose sugar to ketose sugar. This is in aldose form and this is in ketose form. Fructose is a ketose sugar, glucose is a aldose sugar. So aldehyde is converted into ketone here. So the conversion of aldose to ketose form, isomerization. Glucose 6 phosphate is isomerized to fructose 6 phosphate. The first reaction is phosphorylation reaction, where glucose is phosphorylated to glucose 6 phosphate. Here one ATP is utilized. The second reaction is the isomerization reaction, where glucose 6 phosphate is isomerized to fructose 6 phosphate, and this reaction takes place in the presence of phosphohexoisomerase. So in the next reaction, this fructose 6 phosphate, again it undergoes one more phosphorylation reaction. So this fructose 6 phosphate is again phosphorylated by one ATP molecule, and this reaction takes place in the presence of phosphofructokinase, and the resultant molecule formed is fructose 1,6 bisphosphate. That is, one phosphate molecule is attached at the first carbon, and one more, the second phosphorus molecule is attached at sixth carbon. We have six carbons in fructose and glucose. So first carbon has one phosphate molecule and sixth carbon has one phosphate molecule. So this reaction takes place in the presence of phosphofructokinase. By the formation of this fructose 1,6 bisphosphate, the first stage is completed. Okay. Then the second stage. The second stage is formation of pyruvic acid. So the major step in the sequence of reactions is the cleavage of fructose 1,6 bisphosphate into uh, two 3-carbon compounds, say triose phosphate molecules. This is glyceraldehyde 3-phosphate and this is dihydroxyacetone phosphate. And this reaction, this splitting reaction or cleavage reaction takes place in the presence of the enzyme called aldolase. This is a 6-carbon compound. It cleaves into two 3-carbon compounds. One is glyceraldehyde 3-phosphate and the other one is dihydroxyacetone phosphate, and this reaction takes place in the presence of the enzyme called aldolase. Okay. Again, these glyceraldehyde 3-phosphate and the dihydroxyacetone phosphate are isomers, and the further reactions they take place only in the form of glyceraldehyde 3-phosphate. DHAP won't undergo any further reaction, so it has to be isomerized to glyceraldehyde 3-phosphate to continue the further reaction. So it undergoes isomerization, and now we have two molecules of glyceraldehyde 3-phosphate. This dihydroxyacetone phosphate isomerization undergoes isomerization in the presence of triose phosphate isomerase enzyme. Triose phosphate isomerase enzyme. And now we have two molecules of glyceraldehyde 3-phosphate. Okay. So uh, now this glyceraldehyde 3-phosphate, it undergoes oxidation and a 1,3-bisphosphoglyceric acid is formed. Okay, this is the first energy yielding reaction. Okay, this glyceraldehyde 3-phosphate undergoes oxidation, resulting in the formation of, see, these aldehydes on oxidation gives rise to acids. So glyceraldehyde undergoes oxidation, and the resultant um, products are acid, that is 1,3-bisphosphoglyceric acid. Here you have only one phosphate, but one inorganic phosphate is accepted from the cytoplasm, and uh, two phosphate molecules are seen in the this 1,3-bisphosphoglyceric acid, one at the first carbon and the second one at the third carbon. Okay, see here, uh, whatever hydrogen ions are liberated, this NAD, nicotinamide adenine dinucleotide, accepts and undergoes reduction, resulting in the formation of NADH. This is energy rich compound. We have to note all these things. Okay, so one NADH is formed in this oxidation reaction. So this is the first energy yielding reaction of glycolysis, where glyceraldehyde 3-phosphate is oxidized to 1,3-bisphosphoglyceric acid by the action of uh, glyceraldehyde phosphate dehydrogenase, or simply we can write it as dehydrogenase. This reaction involves the incorporation of inorganic phosphate and oxidation of aldehyde to acid. Okay. Here the coenzyme, this is coenzyme, is reduced to NADH. Then in the next reaction, what happens? This 1,3-bisphosphoglyceric acid, now it loses the extra phosphate ion. Earlier it has two phosphate ions. See, you can see here 1,3-bisphosphoglyceric acid, but here it undergoes dephosphorylation reaction. It loses one phosphate ion. Okay, so this phosphate molecule is accepted by ATP, and the formation of ATP takes place. See, yeah, this is the first oxidation reaction where NADH is formed, and this is the first phosphorylation reaction, that is dephosphorylation for 1,3-bisphosphoglyceric acid, and ADP undergoes phosphorylation. Okay, so 3-phosphoglyceric acid is formed and one ATP is formed in this reaction. So, and this reaction takes place in the presence of phosphoglycerate kinase. This reaction is also known as substrate level phosphorylation. It involves the transfer of a phosphoryl group from a high energy compound to ADP. Okay, ADP accepts the phosphate molecule and converts into ATP. This is known as substrate level, as it is ATP is formed during the reactions, the energy released during the reactions. Okay, in here, so it is known as substrate level phosphorylation. If it takes place, if by or phosphorylation takes place in presence of sunlight, or if it utilizes the energy in the presence of sunlight, then we call it as photophosphorylation. Okay, ATP is formed from ADP by making use of radiant energy. Then it is known as photophosphorylation. But here it is known as substrate level phosphorylation. Whatever energy is released during this uh, breaking and making of bonds, that energy is utilized to for the formation of ATP. So this is known as substrate level phosphorylation. So now we have 3-phosphoglyceric acid. So this 3-phosphoglyceric acid is later converted to 2-phosphoglyceric acid in the presence of the enzyme called phosphoglyceromutase. This is a mutation reaction. The phosphate molecule is moved from third carbon to second carbon. Okay. Earlier it was 3-phosphoglyceric acid. Now it is the phosphate molecule moves to second carbon. So it is 2-phosphoglyceric acid, and this reaction is known as mutation reaction, and it takes place in the presence of mutase enzyme. So 2-phosphoglyceric acid, now it undergoes dehydration. One molecule of H2O is removed from 2-phosphoglyceric acid, and the resultant product formed is phosphoenol pyruvic acid. So by losing one molecule of water, a 2-phosphoglyceric acid becomes 2-phosphoenol pyruvic acid, and this reaction is catalyzed by the enzyme enolase. So this phosphoenol pyruvic acid ultimately undergoes the final dephosphorylation reaction, resulting in the formation of pyruvic acid. So in the final reaction, the phosphate group from phosphoenol pyruvic acid is removed with the production of pyruvic acid. Here, the phosphate molecule, which is uh, liberated by phosphoenol pyruvic acid, is accepted by ATP, and formation of ATP takes place. ADP absorbs the phosphate and converts into ATP. Okay, and this reaction takes place in the presence of pyruvate kinase enzyme. So for each glyceraldehyde 3 molecule, we have one pyruvic acid. We have total 2 glyceraldehyde 3 phosphate molecules. That is one from DHAP, which is isomerized to glyceraldehyde 3 phosphate, and one glyceraldehyde 3 phosphate. So we have a total of 2 3-carbon glyceraldehyde 3 phosphates. Each glyceraldehyde 3 phosphate is converted to a pyruvic acid. So at the end of glycolysis, the glucose is uh, splits into 2 3-carbon compounds, that is pyruvic acid. This is a 3-carbon compound, this is a 6-carbon compound. So each glucose molecule, it splits into 2 molecules of pyruvic acid. Here you can see that a total of 4 ATP are formed. See, each glyceraldehyde 3 phosphate, 2 ATP formation. You can see here, 1 ATP here, 1 ATP here from each glyceraldehyde 3 phosphate. We have 2 glyceraldehyde 3 phosphates. So a total of 4 ATP are formed. And already used, 2 ATP are already used during phosphorylation of glucose to glucose 6 phosphate and phosphorylation of fructose 6 phosphate to fructose 1 6 bisphosphate. Already 2 ATP molecules are utilized here. So the net gain is 2 ATP and 2 NADH. See here, glyceraldehyde 3 phosphate is oxidized to 1,3 bisphosphoglyceric acid. During this reaction, hydrogens are liberated, and these hydrogens are used by this NADH, and it undergoes reduction to NADH. NAD is reduced to NADH. One NADH formation for each glyceraldehyde 3 phosphate. We have 2 glyceraldehyde 3 phosphates, so 2 NADH. So the net gain of ATP is 2 and NADH2 during the process of glycolysis. Okay, so this is all for today, and tomorrow we will see the Krebs cycle and electron transport system. Okay, thank you for today.